that it's like they never change, right? But I change often for fun. I mean, hey, it grows back. Okay. So what do you think of the haircut? I did it myself.
Honestly, it looks fantastic.
Thanks. I bought a $25 pair of clippers like a year ago, and I have never gone to get another haircut. It's like $25 every haircut, or every 6 months with buzz in my own head. Okay, anyway. All right, so thank you for pointing out the fact that I look like someone else. And you think I should be a harder grader?
No, not at all. You should stay the
Like
same.
I shouldn't be a harder grader. I just look like I should be a harder grader. All right, well, let's get talking about fungi.
Are we having quizzes Wednesday still?
Well, quizzes are done Fridays.
Fridays?
Yep, so go ahead and look at the quiz schedule. We're still on schedule, right? The next one's set up for the 31st, which is Friday. The materials and methods in my lab are not due this week because we never talked about them. We're going to talk about them this week, and they'll be due next week. Nobody actually has the results yet, so the results are going to get pushed back off as well until we have some numbers. Okay, so we're going to talk about fungi, which is the other major group of multicellular eukaryotes, where you've talked about plants already. You guys talked about protists last term. Those are unicellular for the most part, although there are some multicellular ones. And we'll be talking about animals by the end of the week and beginning of next week. But before we talk about that, we have to talk about the fungi. Fungi are single-celled or multi-celled. It depends on the group. They're eukaryotes. What does being a eukaryote mean? They have a nucleus and they got mitochondria. Right, okay. Fungi are specialized to be digesters. They digest things extracellularly, which is pretty cool, right? Lots of organisms ingest things. These guys digest them outside the cell. They secrete digestive fluids, which break down the things that they're growing through, and then they absorb those nutrients directly from the environment around them. They are either sexual or asexual reproducers. There are some lineages that are totally asexual as far as we know. There are some that are able to go back and forth between a sexual and non-sexual stage. And they've got some really weird sex. We'll talk about that at a certain point. They are more closely related to animals than they are to plants, which is an interesting thing because they had traditionally been considered a type of plant. They have cell walls, right? But their cell walls are made of something different. Their cell walls are actually made of a different material than the cell walls of plants. When you look at their ribosomal RNA sequences and you look at their genetics, they are much more closely related to animals than they are to plants. In fact, when you look at how close they set up, it's only 600 to 700 million years ago that the fungi and animal groups split. So this is like a relatively recent split in the eukaryote line. The diversity of these things is increasing in terms of our knowledge of it in incredible ways now that we have molecular tools. So I had a friend in graduate school who was a fungal systematist. And as a graduate student, right, this is a person who is working on their PhD, he had identified somewhere on the order of 300 or 400 new species of fungi from molecular means. So he just said, he was doing these big screens of fungal samples and said, okay, here's 15 new species in this one sample of soil that we've never identified before. And they're now waiting for somebody to figure out what they actually do and what their names might be. Nobody's named them. And at the end of his 6 years of graduate school, he had hundreds of sequences that represented new species of fungi. This is one guy working in one location, mostly in New Jersey, like where, it's the most populated state in the country. You'd think we'd have a pretty good grasp on the fungi in the most populated state, but not so. The numbers are potentially in the millions. 1.5 to 2 million is not an unreasonable estimate for the number of fungal species that are out there. There are orders of magnitude less than that described. So the number of species in terms of what is out in fungal diversity could be huge. This tree represents the eukaryota. You've probably never seen something like this. Maybe back when you did your who had Raimondi for Protists? I don't even think she used this group. These are the major groups of eukaryotes. The Archaeplastida, that's the plants and green algae. Rhizaria, those are phylopode amoebas. Aviolates, those are things like ciliates. Stramenophiles, amoebozoans. You guys, do these sound familiar at all? From Q100? Depends on whether you were in my section or not. The Epistoconts, this group, is our group. You'll notice here we are, animals. But in our group include the fungi. They're that closely related to us. They're more closely related to us than plants or any of these other groups of protists. So fungi and animals are actually on the same lineage in terms of eukaryotes. Fungi are they're kind of bizarre because they're multicellular, but cells aren't really the relevant way to think about these things in terms of their reproduction and in terms of their development. They're really found as these long, slender threads that are filled with cytoplasm. Those threads are called hyphae. And those threads can have septa or not. So they can be these long, continuous threads that have no breaks between them and might have a whole bunch of nuclei within that one long thread. Is that one cell, or is it a multinucleate cytoplasm? There are some that have little breaks. These are called septa, but the septa have holes that allow nuclei to pass from one to the other through the septa. So they're kind of weird, right? The cytoplasm can flow through septa. Cytoplasm can flow through a hyphae, including the nuclei within the cytoplasm flowing through the interior portions of the hyphae, including moving from one septa to the next. So you see how cells are kind of weird here, right? The whole idea of like an individual discrete cell doesn't really match the way these things grow, because they grow in these long, slender threads that may have or may not have multiple nuclei. So these masses of hyphae get called mycelium. And the mycelium grows through the substrate. As it grows through the substrate, it secretes digestive juices, and those juices break down the material that it's growing through. Their cell walls are not made of cellulose, the things that plants are. They're made of chitin. Does anybody remember back to 200 what chitin is compared to cellulose?
It's like a hard shell.
Well, it can be made into a hard shell. Arthropods have chitin. It's the only other group that does. Arthropods. Chitin. Chitin's similar to cellulose. It just has a different way that the individual sugars are bound together. So it's similar to cellulose, but it's got a slightly different structural, chemical structure. Okay, so what you're looking at here is individual hyphae that's up close, spreading as mycelium through a substrate, and the fungus embeds itself between the cells or even within the spaces of wood or dying material or dead plant material, and digests it from within. Pretty awesome, huh? I just inoculated 50 logs in my basement with fungi. You know how they were out trimming the trees a couple of weeks ago? I asked the grounds guys if they would save me some logs, and they saved me a bunch of logs. And then I bought inoculated little pieces of dowel, little wooden dowels that already have the mycelium grown in it. And you knock the dowels into holes within the logs, and the mycelium starts to grow throughout the logs. And when the entire log is filled with mycelium, they fruit. What you guys think of as the mushroom is really a small component of the organism. The organism is the connected mass of mycelium growing within the substrate. The mushroom is just the fruiting body. And anytime you see a mushroom, what it represents is a much larger mass of mycelium that are utilizing the resources of the substrate it's growing on. So the things that I inoculated are shiitake mushrooms, blue oyster mushrooms, phoenix oyster mushrooms, and pearl oysters. And then I'm going to put these things out in my yard, and when the conditions are right and the mycelium has grown through the log, I'll be inundated with hundreds of pounds of shiitakes. Shiitakes get about $20 a pound. So I basically just got myself a couple of $1,000 worth of mushrooms in my backyard. Of course, it'll take 6 or 7 years for me to get them. There'll be nothing for the first year as the mycelium grows throughout the substrate. But it's a pretty interesting way to live, right? They obtain their food from what it is they're in. So fungi that are living within, like, a dead log are actually growing into those spaces of the xylem and between the tracheads and filling up the locations where the lignin is and where the cellulose is, where they secrete an enzyme that breaks apart those lignin bonds. We can't do that. All animals can't do that unless they have symbionts that are able to do it. Fungi, if it weren't for fungi, we'd be neck deep in dead plant material because they do a great job of breaking it down. So they break down what's around them. And then they absorb those smaller molecules through their membranes and use them as their nutrition source. In order to be able to do that, they have to be able to be in contact with an incredible amount of substrate. So the mycelium, with all these little hyphae growing through everything, makes for a huge surface to volume ratio. That means no cytoplasm is really ever very far away from a membrane that is actually secreting and incorporating and picking up the small molecules that have been digested off of the substrate. They have developed some really interesting ways of feeding. Not just as decomposers, there are actually some fungi that are predators. Doesn't that sound awesome? If you look at that upper left-hand corner, this is a fungi that is a predator of nematodes. It actually grows a fungal hyphae that forms a loop, and as the nematode is moving through the soil, if it goes through the loop, it gets caught by the fungi. And the fungi then secretes a material that digests holds onto and then digests the nematode before it can get away. You have fungi that digest you when you're still alive. Anybody ever have athlete's foot? Oh, come on, don't lie. Thank you. All right? That's a fungi that's digesting you while you're still alive. It's growing into the spaces between your skin cells and digesting your skin cells. I was once in South America when I was in Brazil. I got a fungus growing on my back. It took me 3 years of putting a fungal cream on my back to get rid of it every day.
That's awful.
It was awful, right? But think about it, right? Most drugs exploit something that's very different about the disease you have, right? And fungi are so close to us the distance of finding something exploitable, right? Bacteria is easy. They've got cell walls. We take some antibiotics, make the cell walls not grow, boom, bacteria is gone, right? Fungi are so close to us that it's hard to find a drug that will kill the fungi without killing me, right? That the distance between us genetically is not that far compared to other potential antibiotic type things. So I had to put this fungal cream on my back. It took 3 years to get rid of the damn stuff. If you go to the tropics, that might just happen to you. It's wet, hot. Fungi love that kind of stuff. It's not because I didn't take baths. I know some of you are thinking that. All right, so one of the things that's really cool about fungi is that there are different nucleus numbers. So, okay. In some cases, we have hyphae that can have septa, one nuclei per septa. Those are called monokaryotic. One kind of nuclei found in each septal section of either. There are some that have two nuclei within each septa section. Those are called dikaryotic, two nuclei. Those two nuclei are usually different from each other, but both are being transcribed. So in a monokaryotic, they have only one kind of nuclei, one genome. It gets expressed. In dikaryotic, there's two nuclei found per septa section or per hyphae. Both are being expressed. It's not diploid, even though it has a genetic complement of 2N. Those N's are both being represented, so we call it N plus N. Okay, it's an N plus N. So it's not diploid. They haven't fused yet. We're going to see that they can. But they're both there, both representing two genomes at the same time. So, if the nuclei has intermingling, if a hyphae has intermingling nuclei, they can all be one type if it hasn't fused yet. We'll talk about that in a moment. Those are called homokaryotic. Every nuclei has the same genome. In the setups where there are more than one nuclei type, you can have heterokaryotic, so that you might be dikaryotic and heterokaryotic, two nuclei per septal section. Both are different from each other, and yet both are expressing at the same time. So remember when I said their sex is weird? This is weird sex, right? This is like they're going to have a recombination in sexual reproductive sense, but they actually sort of sit with both types of genomes represented at the same time before they actually fuse. So that's really, really weird. The cell isn't really the best way to think about these things, because in fact, cells don't divide in the same way that cells divide in most other eukaryotes. They don't have mitosis take place and then the cell split into two. Very often what you'll have, and this is an example of a hyphal, of a septate hyphal growth, where a new portion of hyphae grows out past the septa before it's got any genomic material. So you'll see you've got this growth of a new hyphal thread out here. The mitotic spindles form in the new half and pull the nucleus through. While it comes through the septa, the nuclear envelope falls away. All of the chromosomes move into the new half, and then they split in half and move back, half of them move back into the old half. Totally different kind of reproductive, or totally different kind of mitosis than what we see in most animals. What you saw in mitosis back in 200, this is weird. So you get your entire genomic material moves over to the new hyphal thread. then it goes through the chromosomes pulling apart, moving one set back into the old, and now you've got two nuclei, both with the genetic complement. If this were dikaryotic, both nuclei would divide and both would move into the new hyphal part and move back into the old hyphal part. And both would still be being expressed. So you don't need to necessarily know the details of this, but what you do need to know is that its mitotic division is different than what we see in animals and plants. That you still end up getting an exact copy of whatever the genetic complement is into the new cell part, putting cell in quotes here, because really what it is a new nucleus forming. So it's strange compared to regular mitosis. So with the weird reproductive mitosis, with the weird way that they are able to have dikaryotic cells or hyphae, what ends up happening is that they have interesting ways of reproducing. They can reproduce sexually. We'll talk about that in a little bit more detail when we start talking about the individual groups, or asexually. In sexual reproduction, there's two compatible hyphae types that come in contact with each other. So instead of having like males and females making sperm and egg, you have a plus type and a minus type of hyphae that when they meet each other and recognize their compatibility, they fuse together. But the nuclei don't fuse together, just the cells, forming what had been monokaryotic, one kind, plus and minus, forming to make a new N plus N hyphae that is dikaryotic and heterokaryotic.
So do they always start off as monokaryotic?
Do they always do what?
Start off as monokaryotic?
Yeah, and we call it, well, it depends on the group, but we generally call that primary hyphae being monokaryotic before fusing with a compatible type, and then secondary hyphae after they fuse. There might be nothing different about their growth forms or about their habits or anything, but if they're monokaryotic, primary hyphae, they cannot form mushrooms or fruiting bodies. They've got to be dikaryotic to do that. We'll talk about specifics of each group as we get through the moment. So, all right, so we've got some groups that do this immediate fusion, results in a 2N diploid cell. Those are not what you normally think of as mushrooms. We can talk about them in a moment. Others are dikaryotic that have an N plus N stage. Most of the mushrooms that you think of when you think of mushrooms are dikaryotic with an N plus N stage. These that have dikaryotic stages often form a fruiting body, and the point at which those N plus Ns fuse happens only in the fruiting body. So like a mushroom itself, all of the hyphae that are forming the mushroom are N plus N. And those mushrooms that make spores, the formation of the spores is actually the outcome of the reproductive fusing. That would be the karyotomy. All right, so let's talk about spores first, because spores are how.
So if it doesn't create the fruit, it's not going to have the one.
That's right. For at least for the Ascomycota and the Basidiomycota that we'll talk about once we start talking about individual groups. But when you think of a mushroom, what the mushroom is doing is making a location where the two different kinds of nuclei can fuse to form a diploid cell for a moment that then goes through meiosis to make haploid spores, and those spores can then get released. We'll go into the details of it in a moment. All right, so spores, you're going to notice that we are using plant terms here, right? It's problematic because we know they're not actually that closely related to plants, but their history was one of these are a specialized kind of plant. So you're going to see the same terms like sporangium and gametangium and spores, but they're different than what we talk about in plants, even though we're using the same terms. All right, so spores are the main ways that fungi disperse. Spores can be formed either sexually or asexually. Okay, that's weird, right? Where the spore is the outcome of either just an asexual creation of the spores or a sexual forming of those spores. So, fungal spores are mostly dispersed by wind. They're very light, and they're ubiquitous. It's a good word. If you don't know what the word ubiquitous means, it means they're everywhere. They are everywhere. They're in the air around us all the time. So, spore lands in a new location, forms a new hyphae, and that's how they get dispersed. This structure here is a conidia, and that's formed from asexual formation. So each one of these spores has the exact same genetic complement as the parental hyphae, which is different than the sexually reproducing methods. We'll talk about that. All right, so fungi are still an interesting group to be working on. Think about it, right? I got a friend who took a bunch of soil samples and ended up with hundreds of new species of fungi. And these are still being worked out. And in fact, they're different. What I'm telling you now is different than what I learned as an undergraduate a mere 25 years ago. Our molecular tools have altered our view of the relationships of these groups. What we do know is that there are 7 clearly monophyletic groupings. You guys remember what monophyletic means from last semester? You don't.
Oh, no, I'm sorry.
Someone want to take a stab at what monophyletic means?
They're like one big branch.
That they're one branch, right? These represent a common ancestor and all of their living relatives. So each of these 7, clearly monophyletic, represents a single lineage of living organisms that shared a single common ancestor. So these 7 monophyletic groups are the Microsporidia, the Blastolidiomycota, the Neocalamaptygomycota, these are really hard to say, try that one 3 times fast, the Critidiomycota, the Glomeromycota, the Basidiomycota, and the Ascomycota. 7 major groups of fungi. The ones that you think of when you think of fungi are the bottom 2, the Basidiomycota and the Ascomycota. We're going to get to them at the end. You're going to need to know these other weird ones, but the details of reproductive cycles, you're going to need to know some of them. All right, so here's how cool these are. When I learned Microsporidia 25 years ago, I learned it in the protists. It was thought to be a protist. Single-celled. Turns out it's actually a fungi. A fungi who, when you look at their relationships, their genetic relationships, are very clearly grouped with the rest of the fungi. These are all parasites, the Microsporidia. They're all obligate, they are all intracellular. That means they invade a cell of a host and live inside that cell. They cannot survive outside the cell. They lack mitochondria, and that used to be an argument that they represented a very early stage of the development of eukaryotes, a point before mitochondria, but we now can find mitochondrial remnants in their genomes. So even though they don't have mitochondria, they have mitochondrial genes.
So they evolved to not have
They lost them. And if you think about what they do, they live within a cell. There's plenty of glucose around. You don't need to go through the rest of glycolysis or the rest of the Krebs cycle to get enough energy because you're surrounded by the glucose within the cell. The cell didn't have enough glucose, the cell wouldn't be surviving. And so it just does glycolysis to get all the energy it needs because it's got glucose all around it. It's basically abandoned the whole rest of the metabolism that the rest of the eukaryotes have and just said, I'll be fine with glycolysis. But it still has genes that are mitochondrial. So we think that it had mitochondria and lost them. Okay, so they're really, really weird. If you want to know more about them, we will talk about them briefly in invertebrate zoology, my upper level course on invertebrates in the protist section, even though they're not protists. Okay, so the next three groups have a bunch of similarities that we used to have them all grouped together. These are the Cryptomycota, the Blastoclidomycota, the Neocalamastogocota, Mastigomycota. I forgot the one syllable in there. Seriously, say that 5 times fast. No, seriously, do it. Then you'll get used to saying Neocalamastogomycota. Mastigo means flagella. So the thing that all of these guys share is that they have a flagellated zoospore. So what that means is that they have a stage that where they fuse together and make this diploid flagellated zoospore. They all have it. We think that flagellated zoospores were lost in the other lineages, but this represents sort of the ancestral state. They're important because they make they make some diseases. So there are some waterborne forms of these that cause intestinal distress when people get them. There are some that act as parasites on other organisms, like this is parasitic on an algae. This thing actually lands on the side of the algae and secretes digestive juices and then starts to digest the inside of the algae. Frogs and amphibians that don't have a keratinized skin are often susceptible to being infected by these things because they move into the spaces between their skin cells. So this frog is showing these little growths on them, and what those are actually representing is individual Cryptomycota that are sitting within the spaces between cells. And so they're sitting within the cells, and they're secreting digestive juices and digesting the cells themselves.
So it's just eating away the skin, or does it eat away deeper tissues?
It's just on the skin. But if you're an organism like that doesn't that moves gases across their skin, having something eating away your skin cells could be problematic and gross. All right, so those three groups, that's all you got to know about them. We're going to talk about one of these, and its weird life cycle. Where'd you learn this? What's that?
It's like trigger.
Oh, you mean a life cycle? Oh, good God, you people, we are going to go. We've got life cycles of fungi, and when we get to the life cycles of plants, you're going to wish, or when we get to the life cycles of animals, you're going to wish that we still were doing the life cycles of plants. Because in life cycles of plants, a sporangium's a sporangium's a sporangium. When you start looking at some of the other life cycles that we're going to look at, while it goes diploid organisms, haploid gametes, there's all kinds of other weird stuff that take place in there. Yeah, just wait till we talk about the platyhelminths. All right, so this is Allomyces. This is a Basidiomycota, Basidioclidiomycota that is a soil fungi. You'll notice that we're using terms that are very similar to plants. Sporophyte, gametophyte. Sporophyte would have what genetic complement? Diploid, 2N. Gametophyte would have haploid. The gametophyte makes what? Gametes. Sporophyte makes what?
Spores.
Okay, good. You learned something last year. All right, so let's start here with our sporophyte. In this case, we have haploid gametes that fuse together to form a sporophyte. So this is a diploid organism. In this group, it's not N plus N, it's actually 2N. They've form together a diploid zygote that then begins to form hyphae. The young sporophyte gets all these hyphae growing. They can make sporangium that then have diploid zoospores that can swim around and form new sporophytes. So these are diploid zoospores are actually asexual reproduction. Okay, talk about weird. Flagellated cells that are diploid that are asexual. Okay, that's just bizarre. Pretty much every other place we see flagellated anything, we're talking about sperm, or we're talking about haploid gametes. Flagellated diploid zoospore. Weird. Some of the sporangium can make haploid zoospores. Those haploid zoospores develop into gametophytes. Gametophytes are haploid, and they grow up to make male and female gametangia. Those gametangia make both flagellated sperm and flagellated egg. The difference is the size of the cell. Yeah.
So is this a dominant gametophyte or a dominant spore?
Neither. This is a hyphal mass that may be a gametophyte, it may be a sporophyte. The only thing that makes a difference is it making gametangia or is it making sporangia? But they can be equally abundant. Yeah. All right. Okay. Yeah.
So, because of that, are the gametophyte and sporophyte like visually discernible?
No. The gametophyte, they're only visually discernible if you're looking at the sporangium or the gametangia. Right? The gametophyte has two separate looking structures. One, a male or large and small, making zoospores or gametes. The sporophyte has one kind of sporangium, although it depends if it's making sexual or not sexual.
So it's homospores?
Don't use that term.
Okay.
It's got an asexual spore, zoospore, and a sexual zoospore. It can make a haploid or it can make a diploid. So, all right. So, don't worry. That's the least important of the life cycles. Well, there'll be some other ones that are much more important for you to know. This group, the Zygomycota, is sort of a hodgepodge group. Still being figured out. We think of them as being structurally similar, but their genetics are showing they're probably not monophyletic. So we don't really know how this group is going to work out. As we get more data, some of them are being grouped together in some groups, some of them are being grouped together in others. Don't worry about it. Unless you want to go get a PhD, this would be a great place to go look for, go have a career as a fungal systematist. Plenty of jobs out there for fungal, I don't know, we'll see. Plenty of unknown questions for a fungal systematist that can answer. Things like the gray bread mold, that is one of these, a Zygomycota. Rhizopus is the name of that. All right, so here's our Zygomycota. Rhizopus, common bread mold. We're going to start, let's start here with this. Let's start here with the spores, okay? The spores land on edible material. They germinate into a new hyphae. The hyphae grow around on top of the stuff. There are two different compatible strains. So you can have plus type and minus type hyphae growing around. They look the same, you can't tell their difference. When a plus type and a minus type are growing together and they come in contact with each other, they start to form a specialized spot where the two strains come together. and that forms a gametangia where fertilization will take place. So sexual reproduction happens when the compatible named strains come in contact with each other to form a gametangia. Asexual reproduction happens when they don't find each other and they don't make a compatible setup. And they'll form these structures that are filled with asexually made spores. Those asexually made spores can land somewhere else and grow, and they won't go through sexual reproduction unless they find a compatible strain that they're growing next to. So this asexual reproductive can take place if they don't come in contact with a compatible strain. When they do find a compatible strain, they form these little plus and minus type things and form a gametangia, and within the gametangia, the two nuclei from the two different strains combine. So that's your karyogamy step. But the karyogamy step is very short-lived. The N plus N stage quickly forms a 2N stage, and that 2N stage then goes through my meiosis to form an N hyphal setup. The hyphae that are now coming from these spores are haploid. So the diploid stage is only brief. This is called the zygosporangium, where those two nuclei move into the same cell in the zygosporangium, fuse, and then start growing new spores that are haploid. So the diploid stage is very short-lived. You're going to look at this in lab. We'll have the chance to see the karyogamy or the chance to see the zygosporangium and these asexually reproducing bodies, which are called conidia. Zygomycota. Yeah.
So karyogamy is when it's N plus N?
Karyogamy is when the N plus N fuse together. So to form a diploid portion. In this case, the diploid is only for one cell that then goes through meiosis, right? And in fact, in most fungi, that's how it works. You end up with basically diploid for one cell that then separates. If you have two nuclei, you can have dikaryotic, so hyphae can fuse, have both karyotypes present and functioning, but the fusing together to a diploid usually happens in one cell for one nucleotide. Okay? All right, so the next group are the glomeromycota. The glomeromycota you guys already have heard about, right? These are the arbuscular mycorrhizal fungi. Remember, plants have a couple of kinds of fungi that can grow within the spaces of the cells of their roots. The arbuscular ones are ones that have growth that go into the cell wall and actually can grow into the cell. These guys are symbiotic associations with plants. And as far as we know, they can't exist without plants. So this is an obligate symbiosis. Some plants can exist without their fungi, but these fungi cannot exist without their plants. So this makes the transition from plants moving to land. It is this kind of fungi that probably helped that happen, and this kind of fungi has now tethered itself to plants to the point where it cannot survive without them.
Doesn't matter what plants it binds with.
It depends on the arbuscular variety. So there are some that are very species or family specific, and they don't reproduce sexually, so they don't actually move to new locations, which is one of the reasons why if you cut down a forest, remember when we were talking about how if you cut down a forest and try and plant the same stuff back, it may not grow back the same way it did. So think about a pine forest that requires arbuscular mycorrhizal association. You cut away the pine forest, you have those mycorrhizae die, and then try and plant back pine trees without their fungal associates. The fungus isn't coming back. The thing about these things is that they form these huge connected hyphal mats. The largest single organism on the planet is a fungi. Probably in some parts of the world we've never looked, we would find them bigger than this. But the one that is of interest is from central Michigan. Somebody went out and looked at Glomeromycota associations with pine trees in central northern Michigan and found that over a multiple square mile area, the fungi had identical genomes, which means they're all the same organism, connected to each other. One giant hyphal net spreading through the forest. When they estimated the mass of it, it was something on the order of like 600 blue whales. It was like this huge mass of genetically identical hyphae, which means one organism, all connected with all of their neighboring trees. Okay, so they don't reproduce sexually, and they can't survive without their host, the Glomeromycota. Here's how their growth is, right? They don't above ground fruit. They grow into the root, outside of the endodermis, invading individual cells, forming the envelope. They probably are really important for plants getting nutrients from the soil, especially phosphorus, which is hard to uptake for plant roots. They're essentially increasing the surface area that is available to the plant by the surface of the hyphae. Okay, Glomeromycota. Yeah.
Is this the same division that also will like kill plants? Because like, I know some of my mom beans
Are covered in fungus. So we have fungal nets upstairs. And what those fungal nets are doing is they pick up the spores from the fungi that's growing in the soil and then transfer them to the surfaces of the plants.
that doesn't help those.
No, the fungi actually will grow down into the plants and will start to digest the plant cells. So if you have like weird leaf curl on your plants or like sections where there's dying parts of the plants, that's fungi that's been introduced by those fungal nets. You notice my plants up there are also getting affected. When they go out into being put out into the garden, they'll outgrow those infections. They'll still have the fungi, but it doesn't affect yields. Different kind of fungi. That fungi is mostly this kind, the Basidiomycota. There are some Ascomycota that do it too. So when you think of a mushroom, you're thinking of this group. Your experience with fungi is probably mostly with this kind of fungi. If you eat fungi, anybody eat mushrooms? Then you're eating this group. Unless you're eating a morel. Anybody ever had a morel? If you're eating a morel, you're eating a different kind of group. Okay? Pretty much the only one in truffles ends up being in the Ascomycota as well. So, okay, the Basidiomycota are the fruiting fungi that you think of when you think of fungi. They have a fruiting body that has a spore dispersal system. Things like mushrooms, toadstools, puffballs, shelf fungi, those are rusts, smuts. Those are all this group. They are ubiquitous around the world and incredibly economically important to humans. They're named for their reproductive structure that's called the basidium. That reproductive structure comes on this little club-shaped structure called basidium. The basidium is the location where karyogamy happens. So you'll have an N plus N secondary hyphae growing everywhere. When the conditions are right, they put up a fruiting body. The fruiting body is all N plus N except in the basidium. The basidium, N plus N, fuses to form a diploid cell and then goes through meiosis to form 4 haploid spores. So those haploid spores end up being stuck on the end of the basidium. So see this thing that looks like little cow udder? Those are the remnants of what had been where the spores were. So here you see these 4 spores forming at the end of the basidium. Those 4 spores are the haploid outcome of the sexual reproduction, the fusing of the two genomes within the basidium. So that basidium is really the only diploid cell in the entire life cycle. Just one shot. 2 nuclei fuse to form a diploid, go through meiosis to form 4 spores. Those spores are called basidiospores. The mushroom is called a basidiocarp because the location where karyogamy takes place is the basidium. Okay. The life cycle looks something like this. We're going to start with this spot right here. You have a hyphal strain growing around. It is monokaryotic. It's got one type of nuclei. They may be septate, may not be. Single kind of nuclei streaming through the cytoplasm. Comes in contact with a compatible strain. So plus type becomes contact with a negative type. And those 2 hyphal types fuse together to form a single set of hyphae that have both types of nuclei. So you end up with secondary mycelium having N plus N, both genotypes existing side by side, expressing side by side. Both nuclei are able to make proteins. If there's differences between them, you might have 2 types of proteins being expressed in that one organism because both nuclei are being expressed. Grows all throughout the substrate. Only when conditions are right, temperatures are right, moisture levels are right, does a fruiting body form. The fruiting body in this case is a basidiocarp or a mushroom. Mushrooms can grow incredibly fast. You know how it rains and then a day later there's mushrooms everywhere? Well, those mushrooms have already started growing and coalescing into the beginnings of growth forms. But when the conditions are right, mushrooms can appear incredibly quickly. When I go out and look at my mushrooms in my yard after a rain, right about now, like all this rain we just had, I'm sure I'm going to get a flush in the next few days. You want shiitake? Bring it. I actually was thinking I might go home and get the log. I got one log that's got a couple of mushrooms on it today that I bring in. But I don't have a car. So tomorrow, who's in lab tomorrow? Maybe I'll bring it tomorrow. Bring my fruiting shiitake log from my backyard. Okay. So they fuse together, grow all over the place, N plus N. Secondary mycelium starts to grow the basidiocarp. Basidiocarp grows up fast. On the bottom structures, there's usually gills, sometimes pores. In those locations, the basidia line the inside of the gills. So have you ever, who's eaten a regular old mushroom from the store? You look underneath, you see those little lines, right? Have you ever taken a mushroom and just left it on a piece of paper? And you come back and look at it after a couple of hours, and there's a dusting of spores sitting on the piece of paper. It's called a spore print. You can actually use it as an identification tool. Those gills are all lined with hundreds of thousands of little basidia. And inside each of one of those basidia, the 2 different types of nuclei fuse together and form a diploid nuclei. That diploid nuclei immediately goes through meiosis, forming 4 basidiospores that are each haploid. Those basidiospores are blown away, land in the soil, germinate, form a new hyphae. And that new hyphae has one type of nuclei and will, until it finds a compatible strain, when it'll fuse together and form a dikaryotic and heterokaryotic secondary hyphae before it ends up going through sexual reproduction in the mushroom.
Are they neutral strains until they find a compatible
No,
mother
the
strain?
strains are set, right? It's like if you think about there's proteins on the surface of the cells that are recognized by the other cell. So those proteins are already set as soon as the hyphae start to grow. They're set in the genome that is the monokaryotic spore. And so those spores, when they grow, they'll grow if they only find other same type without a compatible cell surface protein marker, they won't fuse to form a dikaryotic hyphae, secondary mycelium. They've got to find one that's compatible. All right, you guys appreciate mushrooms more now, or are you like, holy crap, more like something?
Appreciate them just as much as plants now.
Oh. Thanks. They're way cool.